We were here last year, so really what I plan on doing is just giving you a quick overview of our company and then just telling you where we've come since we were here from last year. But effectively, there's three parts to B-Mogen. We do fee-for-service work where we do gene knockouts and gene knock-ins for therapeutic companies researching various drug discovery kind of things, and that's effectively how we make our living on a day-to-day basis. But then we also, on the your left-hand side, we do a non-viral gene delivery. So we're able to deliver CARs and TCRs permanently into the genome to kind of replace lentiviral transduction methodologies for the CAR and T-cell therapy world. And then lastly, we actually have some expertise at our company. We're the first company in the world that has figured out how to engineer the mitochondria genome. And today we're working on building mouse models that effectively replicate various mitochondria diseases, and then we hope to eventually be in a position where we can correct mutations in the mitochondria genome and add some therapy to our company. So today I'm going to focus on the non-viral gene delivery and what we do in that space. Since we were here last year, at the time we had demonstrated that we effectively had inserted generic cargo in at kind of 30 and 40% rates into effectively T cells, and we were chasing two proof of concept companies at that time. Where we have come to today is effectively we now have actively working on 9 proof of concepts. One of those companies already has a commercial product in the marketplace. 5 of those companies have clinical products in the marketplace, and then 3 are preclinical. We currently are able to do this across a broader spectrum than just T cells. So we can do gene delivery of CARs and TCRs into engineered NK cells. We're working with MSC cells to engineer those cells for therapeutic use. And then we've also done some work with some CD34 cells. And then finally, in one of our proof of concepts, we have gone a head-to-head comparison with lenti viral to kind of make sure that we can deliver on a comparable basis. And when we've used an MOI of 10 on a lenti transduction, we had very similar results. And then additionally, we are working on kind of completing our CMC data safety package for various therapeutic companies that may want to take us into the clinic. So we have just finished all of our copy number studies. Those will be out here shortly in the next week. And then we have site integration that should be done in the next 2 to 3 months. So it's our plan that by midsummer, we will actually have a very robust package that any therapeutic company could take into their CMC section of their IND. And then one of the benefits in working with B-Mogen is if you're a new company and a biotech company, you're trying to get your particular CAR, your TCR put into a lentiviral package and all of that stuff. Typically what we're hearing is that it takes up to a year to kind of get into that manufacturing slot with these various lenti companies and get GMP product. Well, we can effectively do that in kind of between 8 to 10 weeks. On some occasions when it's a harder kind of CAR construct, a multi-dimensional CAR, it might take us 12 weeks, but we can do that easily and very quickly with our technology. So next I want to just kind of go over some of the performance metrics of our platform, and it's a transposon and a transposase system. And again, I want to make sure that you understand it delivers permanent gene delivery. So we can effectively, in generic cargo, we've been getting up to 45% integration efficiency. And then with a variety of in these proof of concepts of CD20, CD19, some TCR projects, we have been consistently in the 20% range of insertion efficiency. We actually have 2 data points that we just got as of yesterday that we were able to put in a variety of CARs in at a 40% efficiency rate that are pretty large cargo CARs, so we think that our platform is very robust. Then we have some proprietary handling methods that we've developed at B-Mogen, so we're able to get high viability after electroporation, and we're kind of in the 75 to 90% range. We don't have any problem of getting to patient dose levels because we're able to get 10-fold expansion. Another benefit that you might find in using a non-viral system, especially a transposon, is you don't have cargo size limitation. So we've been able to prove that we can get in 7 kb of the cargo, not just the whole package, but the cargo into the T cells without a whole lot of degradation and insertion efficiencies. We have demonstrated that we've been over 10 kb, but there is quite a drop-off of insertion efficiencies. And then, as I said, we just completed our copy number. We're kind of in that 2 to 3 copy number range, so we're well within the limits of what both the FDA and the EMA look for. And then finally, one of the benefits about our engineered cells is they are T-stem memory cells. So there's been a lot of data that shows in the literature that shows those are more effective in therapeutic cargoes. And then another benefit of our system is if you decided you were going to do a combination of a knockout as well as a gene insertion, you can do that with one manufacturing step. So those are just the real quick highlights of B-Mogen. And with that, I guess I'll sit down and walk through some questions with Rennie.
Great. Thanks, Jeff. So, we have a mixed audience. We have investors. We have potential partners and companies. I guess maybe taking from the investment side, how do we look at these performance metrics and when we're comparing it to viral-based, metrics? Is it in line, slightly below, actually better? And I guess most importantly, how do these advantages translate into a competitive advantage, whether it's pricing or, delivery?
Sure, great question. I assume I'm on here. So from a standpoint, if you were to compare us head-to-head all the time against Lenti, there'd be a few times when we're going to be equal. And I would say there's many times, just in a transparent way, that we're probably about 80% of the integration efficiency of Lenti. That said, the benefit is, and that's without selection, so that's no selection method on our part. The benefit is our research indicates that the genetic modification process using Lenti at a per patient cost is between $25,000 and $35,000. And we know that they're trying to drive that down into the $10,000 range. We're well below the $10,000 range. So there is a compelling economic argument why you might want to use a transposon system.
When we talk about the 9 partners that you're dealing with right now, all in kind of various stages, but they all seem to be in the similar stage in terms of evaluating your technology. How did these deals kind of come about? Can you talk a little bit about, how many companies you had to talk to get these 9? What's been the biggest pushback, and how do you respond to that pushback?
Sure. So we've taken an approach, being the new kids on the block, that we really need to be compelling for companies that want to try this. So I think we've spoken to 12 companies, and it's been through presenting at conferences like this. So we've gotten people to sign on with the proof of concept, and our proof of concept is very simple. You pay us $50,000 up front. We'll construct your CAR, your TCR, and put it into your therapeutic cells of choice. And if, in fact, you take us into the clinic, we'll credit some portion of that $50,000 to your working with us. And we're obviously looking to get into the clinic to solidify our platform. And so of the 9, really, we've only talked to 12, and everybody has viewed it as a compelling reason to try and find a more cost-effective solution and a more consistent supply chain solution to virus-based methods.
So next year, when you're back at the conference and you're talking about, hey, out of these 9, all 9 or 6 out of the 9 signed on, what does that next phase look like to you financially, as well as how does that translate into how it looks to you commercially?
Sure, great question. So today, out of the 9 proof of concepts we have one partner who has already negotiated a license agreement with us, and we would be a fixed supplier of clinical grade reagents for them for the first 3 years. And there's some royalty-based payments in there as well, some milestone payments. The other people are just starting to engage with those conversations now that we're delivering data to them that shows that we're getting their CAR, their TCR into their therapeutic cell. So we expect, again, we believe that we can be well below $10,000. And, when we think we go through with clinical customers that are ultimately going to go to commercial, we envision them treating 10,000 patients per year at kind of call it $7,000 to $8,000 per patient. And we think that will help grow our company quite significantly.
So let's explore then the business model perspective. You have a for-service business. You obviously have these partnerships as well. Can you talk to us about the potential revenue stream that you're seeing right now? Is that enough to just keep the company self-funded, or would you require some additional capital to transition to a partner-based model?
Yeah, super question. So today our fee-for-service business basically pays for the 15 full-time scientists we have, and we have 6 PhDs that work for us. What we know we need to do is to complete this data safety package as well as build a GMP compliant environment where our customers can buy GMP compliant clinical grade reagents. We figure that investment is somewhere between $6 and $8 million. So we're anticipating we may look at a Series B in the summer time frame, or if we work with the right clients, there might be funding on their sides to get us to that level.
And then I guess finally from a regulatory perspective, what's involved in kind of getting the agency comfortable with a new potential transposon to approve?
Yeah, so we have the benefit of Sleeping Beauty already being endorsed by the FDA and in the clinic. Unfortunately, that's locked up with a single company. Sleeping Piggyback is also in the clinic, so there are 2 transposase systems in there today. Our system is another transposon system, so we think there's a lot of predicates that we can work from, as well as we are building this very robust data safety package, and there's all kinds of reasons why it has safer insertion profiles than virus-based. So we feel pretty good about where we're at and are currently working to kind of get that package to the level where we can have those discussions with the FDA or our clients can on our behalf.
And I guess just one final question for me is, the business model is different than what a lot of other companies pursue, right? They have a particular platform, they apply it to their product, and they move forward in a particular space with their own therapeutic. Yours is more, to give it to anyone, who wants to have it. What was the kind of thinking behind that, as you were developing the company?
Yeah, so when we look at kind of the 2 transposon systems that are locked up, we think today that, every therapeutic company that we talk to is looking for a more cost-effective solution. So we believe a kind of, this is a non-exclusive license that can be used by many is much better for the industry. We will entertain exclusive license for certain cell types or certain fields of use, but a more broad approach we think will benefit the industry better, and we think there's a significant growth in our company if we take that approach.
And I guess I keep saying finally, but this is my real final question. Are there any other areas that you could potentially apply this technology to, right? So we look at cell therapies, pretty much, but is there anything else that either companies are coming to you with or that you're thinking about where this technology could be of use?
I mean, it's in a similar vein. Certainly the people we're working with today are more in the oncology space, but we have started to have discussions with a couple autoimmune disorder companies, and we do think that there's a variety of ways, and certainly looking at the last 2 presenters, there's opportunities that probably go beyond of how we might partner with a synthetic gene company to do yet new and novel things.
Terrific.
Thank
you,
you.
Jeff. Thank you very much, and please thank the presenter.